Hello friends, welcome to some biology class. Friends, today we shall discuss about the alternative pathway of complement activation. This pathway is independent of the antigen-antibody interaction. Because of this, like the lectin pathway, this pathway is considered as a part of the innate immune system. But unlike the lectin pathway, the alternative pathway uses its own C3 convertase and C5 convertase complex. In the alternative pathway, the C3 convertase is composed of one molecule of C3b and one molecule of Bb. This molecule is unique to the alternative pathway, and the C5 convertase complex is formed by the addition of a second molecule of C3b to the C3 convertase. The alternative pathway of complement activation is initiated in three different ways. They are: number one, the alternative Tickover pathway; number two, alternative properdin activated pathway; and the last one is the alternative protease activated pathway. First of all, we shall discuss about the alternative Tickover pathway. Following this, we shall also discuss about the remaining two. The alternative Tickover pathway of complement activation utilizes four serum components. They are C3, properdin, factor B, and factor D. The term Tickover refers to the fact that C3b is constantly being made and spontaneously inactivated, thus undergoing a tickover. The tickover pathway begins with the spontaneous hydrolysis of C3 at its internal thioester bond. This process yields C3H2O. In the presence of serum magnesium, C3H2O bind with a serum protein known as factor B. By binding with C3H2O, factor B becomes susceptible by hydrolysis by a serum protease known as factor D. So, the factor B is hydrolyzed into two fragments. Smaller fragment is Ba, which diffuse away, and the larger fragment is Bb, which remain bound with the C3H2O. This complex of C3H2O-Bb is known as the fluid phase C3 convertase. The fluid phase C3 convertase is also known as the initiating C3 convertase. This enzyme cleaves many molecules of C3 into two fragments. Smaller fragment is C3A and the larger fragment is C3B. This initiating C3 convertase is constantly being made in plasma, cleaving a few molecules of C3 and is then rapidly degraded. But when there is an infection, the C3b molecules bind with the nearby microbial surface via thioester linkage. These membrane-bound C3b molecules bind with the serum protein factor B. By binding with the C3b, factor B becomes susceptible to cleavage by the factor D. So, factor B is cleaved into two fragments. Smaller fragment is Ba and the larger fragment is Bb, which remain associated with the C3b to form the membrane-bound C3 convertase. So, the membrane-bound C3 convertase is composed of one molecule of C3b and one molecule of Bb. After that, the membrane-bound C3 convertase take over from the fluid phase C3 convertase and become the prominent enzyme complex. So, in the alternative Tickover pathway, there are two C3 convertase. One is a fluid phase C3 convertase that kicks off the pathway, and the other is a membrane-bound C3 convertase. The membrane-bound C3 convertase is unstable until bound by a serum protein known as properdin. This protein is also known as factor P. The properdin stabilized membrane-bound C3 convertase cleave C3 molecules and generate a large amount of C3b molecules. The C3b molecules bind with the factor B, facilitating their cleavage that leads to a rapid amplification of the generation of C3b molecules. As in case of the classical pathway and the lectin pathway, in the alternative Tickover pathway also, C5 convertase enzyme complex is formed by the addition of a second molecule of C3b to the C3 convertase that result in the formation of C5 convertase. The enzyme C5 convertase cleave the component C5 into two fragments. Smaller fragment is C5a and the larger fragment is C5b. As in case of the classical pathway and the lectin pathway, here also the C5b molecules goes on to form the membrane attack complex along with the complement components C6, 7, 8 and 9. So friends, this is about the alternative Tickover pathway of complement activation. In the next video, we shall discuss about the remaining two pathways of complement activation.